Hello, and welcome to today's UCSF Virtual Alumni Program, AI and Alzheimer's Research. My name is Leanne Muchler, and I'm a Director of Alumni Relations at UCSF. We hope you enjoy the conversation. And now please allow me to introduce our speakers. Our featured speaker today is Michael J. Keiser, a PhD of 2009, and he currently leads a lab that investigates how drugs affect networks via computational predictions paired with experimental testing. Our moderator, Feng, PhD of 2007, studied in the same graduate lab with Dr. Keiser before joining Calico Labs. Today, they will discuss the interaction between computational science and healthcare and how it is leading to breakthrough innovations for diseases that impact health globally. Welcome, Drs. Feng and Dr. Keiser. The virtual podium is yours.
Thanks, Leanne. Thanks, everybody, for coming to this event. My name is Feng, and it's my pleasure to be here today to moderate our discussion with Professor Mike Keiser. As Leanne mentioned, I got to know Mike when he was a graduate student at UCSF. We were both doing our PhDs in Brian Choiket's lab here, and I definitely remember Mike joining. And I remember his research, even at the time, was really feeling like a new direction for the lab, and it was super fascinating. Mike brought a lot to the lab, a lot of interesting things from his computer science background, math, statistics, and computational approaches that I hadn't been exposed to previously. And though I was a bench scientist, I became really fascinated with the computational approaches that Mike was developing in his thesis project. And I learned an appreciation for computation and data science that continues to this day, where I still work closely with data scientists and machine learning experts in my current work. And it's exciting and fun to think about how to bring these sorts of approaches to bear on complicated biological problems and diseases that all of us are facing. So Mike, thanks for coming today. I'm excited to be able to talk to you about AI and Alzheimer's and other neurodegenerative diseases. And maybe just to get started, you can kind of catch me up on what you've been up to. It seems like you've been publishing a fair amount in the space of Alzheimer's research. So maybe you can start off with telling us what you've been learning there.
Well, thanks, Brian, and thank you for the very kind intro on that. It's good to see you again. So let's see. So from us as a lab, we came at this actually originally from a very more computer science and even chemical informatics sort of background. And about 8 years ago, when our lab started, we were really new to this entire space of neurodegeneration and Alzheimer's disease. And it's really been quite a journey. It's a field that has so many interesting things that are happening right now, so many changes that have happened really recently. So for us, we have gone after a particular slice of it so far. And then over time, we're kind of broadening bit by bit as we learn. But we started with molecules. It's something that you mentioned already, starting in Brian Choiket's lab. And so one of the big questions we have is an atomistic question, which is thinking about small molecules, drug-like molecules. How are we going to even find ways to interact with the proteins that matter for these diseases? Because on the one hand, you have proteins that are things that are directly associated with the disease, as far as we know. And there's been all kinds of cool research coming out there, thinking about these aggregates, thinking about prions, thinking about the propagation of them, and trying to understand how small molecules in three-dimensional space can actually interact with and block these proteins. But then as well, why are these proteins a problem when they're natively occurring as it is already? More often than not, they're not even mutations, but rather this really interesting thing, a structurally misfolded form of a protein. So clearly there must be cellular machinery that usually accounts for this and deals with this, and then over time is going awry. And so that's a whole separate other category of protein we think about when we're coming at this from the small molecule drug discovery side. There's a lot more there to talk about if you're interested in how AI intersects with this and how you even represent both proteins and small molecules through the lens of a machine or deep learning model, because it's not obvious how that would be, because so much of the ways that we think about things that we can often succeed at with these techniques relate to sequences or other information where there's a natural flow. When I speak, the words follow a pattern. When we look at DNA, we think about the sequence of DNA. There's something canonical there. And yet with the small molecule, it's architectural. It's not even obvious how you'd represent that. And so that's one big question for us. But then we kind of go up this pyramid of organization. And we're curious about going beyond simple chemistry or biophysics and thinking about how to go after the disease state. How do we model the disease state when so many things can go wrong? When you think of Alzheimer's disease, it's probably easier to think about the way we think about cancer. There isn't one type of cancer. There probably is not one type of Alzheimer's disease. It's something that we see and we observe at a high level, and we're just beginning to tease out the differences. So if that's going on, how do we come at this without building too many biases into it ahead of time? And bias is a balanced word because on the one hand, bias is expertise, but on the other, if we want to do drug discovery, we have these questions then of thinking about what the mechanisms are that we even go after. Which of those proteins, and how do we leverage data from these screens that are already done, and actually empirically? How do we pull data from that? And so we have questions there about image analysis, about looking at live cell imaging, about thinking about what can we see in these cell systems that model the disease. And we know we can see it, but we have trouble getting the computer to pull that out. That's one category of question. But then the other category too is what can't we see? What didn't we think to look for? We have all these data. Can we find patterns there that aren't already evident to the human eye, but once we know the model uses them, we can dig into that and understand the biology behind it. So we have projects digging into that type of imaging. And then finally, kind of third, in this sort of moving up that pyramid of closer and closer then to thinking about how it's going to come to a treatment, then we think about it in the human. And that's super hard. That's a very difficult situation because already it's difficult to even assess the amount of disease sometimes. How do we bring it in a way that fits to these other parts? And so we've had some really exciting collaborations in the pathology space, specifically neuropathology. Some amazing collaborators there, where we're both figuring out ways to be kind of a force multiplier for neuropathologists, but then also trying to bring a lot of diverse opinions in the community together to bear on understanding these actual pathological hallmarks of these proteins that build up. Amyloid beta protein, tau protein, and others. But then there's this whole other part too. How do we loop that all the way back to earlier on in screening, to earlier on in understanding? What data can we collect when you can still do something about it? Because we need those endpoints. We need to understand what the outcome is. But also, I think, I don't know about you, but I would like to know much earlier what's going on. And so we're just getting into that space right now. And this has been a progression almost linearly in the way I've been describing, kind of upwards from the atoms and the atomistic view through these cells and then tissues and eventually to systems and now to diagnosis as well as where we'd like to go. But that's one of our newest sort of areas. Anyway, so that's kind of a path for us so far, but always coming from this machine and deep learning side to understand how can we augment what we know how to do, but then also how can we kind of open up new areas for discovery.
It almost sounds like there's in the progression that you described, there's probably some exploration that's happened in the tool space too, right? I mean, you start from a place where if you're thinking about atoms, I mean, we feel like we understand atoms a fair amount, but in your pyramid, you kind of got to increasingly complicated problems. And has that kind of, steered the technology development that you've been exploring and the types of approaches that you've been using?
It's kind of a funny thing because sometimes as we climb this pyramid, some of the tools that we use are actually more established outside of academic research. And so when I start at the bottom of this pyramid, all the way down when we're looking at atomistically, we have a pretty good understanding of how individual atoms work or small groups of them. It turns out that really effectively modeling these systems is hard. It's really, really hard. And there's a lot of compute that goes into that. And even then, you have to really constrain the problem. So often that the very best ways to computationally model a small molecule are ways that are incompatible with the sizes of the molecules we care about, much less with the actual cavity or site of the proteins in question. And so we have to get really creative and think about really different techniques to do that. And we steal ideas and borrow them and try to feed them back into the community, from the open source space to do this. Things like graph neural networks, which were originally designed more for social networks. Turns out is a nice way to think about the way that atoms relate, both in two-dimensional and three-dimensional space. And so that's down in the atomistic space. As we go up that pyramid, we begin to think about image analysis. And it turns out so much of the advances in machine and deep learning were done in the image analysis space because there's so much image data. on the internet. And it's a space where the data sets are so public and available. And furthermore, something that we can't ignore is that humans have really good intuition about images. You kind of know when the model is right or wrong, and you can open that box and ask it for a rationale. And then you can assess when it said that's a cat, is it saying it for anything close to the right reasons in the image about what catliness is? Is it even on the cat? Often for the atomistic view, we can get these really detailed rationales that come out of the model, but it takes a lot more effort then to understand if that actually fits with our biophysical intuition. And so there's that. But then finally, when you move all the way up to the what we don't know we're looking for yet, that's when it begins to feel familiar again in that discovery space, in that space of, okay, now we need to really push the methods. And for us, one of the methods we're trying to push in use and trying to also stress test and break if we need to, because we need to know if they're broken, is these interpretable AI methods or explainable AI methods. The jargon is also feature attribution. But getting into understanding if the model is right for the right reasons, we have to really design experiments to think about that if we don't want to rely on confirmation bias or just human intuition.
Maybe to bring all of the audience up to speed a bit, because we talk about this interpretability challenge in machine learning a lot too. Do you want to maybe give the audience kind of a ground up view of the challenge here and what we're talking about?
Yeah. So I think a lot of people have heard about neural networks and about AI in general, machine learning, deep learning, any of these methods where you put up the beginning of a model that can learn basically a very complex mathematical function, or even the very beginning of its own program from a lot of data that you provide. And the critique and the worry about this sort of approach is that it's not based on first principles, in the same way that if you're going to, in a kind of ball and stick way or some other physical way, model atoms or model a system, that you have some basic axioms that you're coming in and saying, we expect the world to work in the following way. And you're baking that into a model, and then you're using that to make calculations and predictions. In this other approach, the models are learning from the data you provide. The critique then is, how do we know that what it's learning has any use? The model is a black box, is what you will often hear, that you can't see inside the box, that we give it data, we give it examples, we train it, and it gets better and better over time at predicting when we ask it questions that we know the answer so far and it doesn't. So we know it seems to be working under our definition of working. But the problem with that is we didn't make the formula. And if you have it show you the formula, it can be giant sometimes. You can have hundreds of thousands to millions of terms in a mathematical equation that the model has learned. And how do you know if that's going to work? How do you know if it's right for anything close to the right reasons? So these are some of the questions and longstanding critiques of AI and machine learning and deep learning. And if it's something that is not a high stakes scenario, that as long as it's right sometimes, and that sometimes is valuable, then you don't need to know in that case. That's good enough. If it does a good job at recognizing different objects, and it does it effectively, and when you do a Google search and you can find cats versus dogs versus airplanes, you might miss some cats, dogs, and airplanes that are unusual, but that's okay. But when we move to a medical scenario, when you're thinking about neurodegeneration, when you're thinking about someone's life, it matters. And so this matters both in the discovery context, because we could try to go down a whole road of discovery based on model predictions that turns out to fail, because what the model learned was a shortcut about the information it gave it, but not something that relates to the broader world. That would be one way it would fail. Or there's other kinds of cases where the model fails, where it's over or underconfident. And we've learned to trust it over time because we were told we had to trust it or not, kind of yes or no, black or white. And in those scenarios, we can be led astray. And furthermore, we don't even know what needs to be fixed in the model too. We don't know how good we are. And so these are some of the motivating questions of this interpretability question. That's why we care about it so much, because we didn't build the reason in. We just gave it a lot of information to get a pattern from. We need good ways to pull out the reasons. And even if it can't tell us the reasons in a human interpretable way, we need good ways to test the reasons so that some of the standard ways that we know could be bad reasons, we at least know those aren't occurring. And so that's this motivation for it. The techniques for it, there's actually quite a few. And it's really fantastic because this is a field where when someone develops techniques for machine learning, for a neural network, for deep learning, for saliency analysis, which is one type of interpretability technique, they put the code online for free. That's baked into the community. And this is a community, this comes from the tech sector, this comes from computer science research, comes from all these conferences that happen in the space. And so then there's this really nice cross-pollination, and we use and apply those methods. One of the really exciting things for us in the space of thinking about Alzheimer's disease is often we run into problems with those methods that aren't obvious from the small test data sets that would be accessible outside of the field. And so that's just one of the things that I think helps us come in as a stress testing use case, where we can find when you take this technique that's supposed to work and we apply it now to really trying to understand something about cellular images being different in the presence or absence of tau protein, and whether the puncta that you see are where they should be, these little intense regions of fluorescence that correspond to these aggregates that spread from one cell to the next. We can actually use the calculations on real medical or drug discovery-based data to test whether these methods to understand the model are even telling us anything different from two different models. And so anyway, I'm going to pause there, Brian. I want to see, am I going down the road you were thinking of an answering that, or have I gone maybe a little too far into?
Yeah, no, I think it's on point. I think there's a lot of, what you described is very general at this point. I think the applications of really powerful technologies that have been developed in contexts that are, like you're saying, a bit lower stakes and a bit more data rich, things like consumer images, optimizing chocolate chip cookie recipes. It's really kind of amazing that someone like yourself can come into a biological problem space and say, oh, I could use a similar approach to solving for an optimum cat recognizer or chocolate chip cookie recipe maker, to understanding how neurodegeneration is happening in the context of a disease. And but if I'm understanding what you're saying right, the weaknesses of the methods that you can address in a lower stakes environment where you understand the data, they're not quite there in these very kind of sophisticated disease settings. We don't have as much data, and we don't have as much native understanding of what's going on to know if the models are on track or not. Did I get that kind of right?
Yeah, and let me try to be concrete about one or two examples. And I also, looks like we have a question in the Q&A too that maybe will speak to this. Okay. So say that we have a neural network, and the neural network is able to predict the binding of a small molecule to a protein that we care about, maybe one that's part of protein quality control. And when we think that this protein quality control, protein's kind of out of control, it's not doing what it should. And so we're looking for drugs against it as a way to get at this question of the mechanisms for a neurodegenerative disease that perhaps is building up proteins that have not been in any way managed, and they would otherwise be dealt with by this first protein. And we have the beginning of a molecule. Something that looks like it almost would work, but it's not going to be a drug yet. It's not good enough. It doesn't fit well enough into the site of the protein that we care about, that we want to change. So we know that from the model because it can score it. That's an example of this neural network doing a good job. It's taken in some representation that we built the best we can in order to predict how good a partner that small molecule from the discovery library is against that protein. The neat thing that opens up interpretability is that we can actually use methods to pretend to train the model still, but instead of training it, what we're actually asking it to do is to look back at that small molecule in the presence of the protein, and we're asking it, how would you change this molecule? Which patterns would you increase? Which patterns would you decrease? Which bonds would you add? Which non-bonded interactions would you remove in order to make the score better? And so from the model's perspective, it's just saying, this is an all right molecule, but it would be better if we had 2 more carbons over here. And that actually becomes a prediction at the same time, one that we can test And we can use other techniques to score it computationally. And we can even go and imagine making that molecule if it's one that's synthetically tractable. And so by doing that, we're combining this interpretability, which was asking it, what would you have changed? And that's following on the technical side. So if you have the score, it's a scalar value, you're following the gradient of it all the way back down into the feature space. And instead of changing the model, which is how neural networks are trained, you're trying to change the data, which is kind of a weird thing to do, but it's changing that protein, that small molecule in the protein. And if you could do that, well, then now that would make it a better binder. And that's the model's hypothesis, which we can go and test. Okay, so that's an example of saliency mapping as it relates to drug discovery in the structural context. You could do a similar thing with images. Which pixels would go up, which pixels would go down in this image of cells in order for this to be a better example of a healthy state. Now, I see that the question we have is there any reason that Alzheimer's as a disease is a good use case for leveraging AI and machine learning as compared to other diseases? And one of the things, there's probably a lot of good answers to that, but for me, something that I find exciting is a term that people are kind of throwing around in the jargony way that's multimodality. And this multimodality characteristic, as it relates to Alzheimer's disease, makes it really nice as a use case for machine learning. And so what is multimodality? It's this idea that for the same sample, maybe you have a tissue sample from a patient. And this tissue sample, there are different ways we can look at the sample. You can imagine if you physically have this sample that you could put it in pathology. You could look at it under a microscope. You could look at a whole slide image at say 40x magnification, and you could, I don't know, do a H&E stain or go actually perhaps for an immunohistochemical stain against amyloid beta. And you can get information about that way. Simultaneously now, though, it's more and more possible from that exact same piece of tissue that is imaged in this way to then begin to pull out sequence data, look at a molecular signature. Additionally, in a way that's kind of indexed or as a grid across the image. This is called spatial transcriptomics. And so now, for the same little bit of tissue, I can see very different types of information. And we care about this in the context of Alzheimer's disease because these are different both hypotheses and different lenses into what could be going awry in the system. And one of the neat things is that we can now even use these two different views, one that has to do with molecular expression patterns, gene expression patterns on the one hand, versus morphology as has traditionally been done in pathology imaging on the other hand. And we can imagine asking models to look for discrepancies between those two things across data set scales that humans couldn't possibly do. And so the models can train themselves to cross across these modalities of information. And when we do that, then we can begin to look for sub patterns within it, sub clusters. And so Alzheimer's fits really nicely with that because as far as we can tell, there are multiple mechanisms. There are some cases where you have examples of this that are genetic, and that's fairly rare, compared to sporadic, which is most common, but is also really frustrating because it doesn't seem to have obvious signals early on. Are there sub-patterns within that? And so that gets into all these other fields in AI and machine learning where there's a real need for us to find this. And so you have fields of this, for instance, unsupervised methods like autoencoders and asking if we can tease out the space based on how much variation there is, all the way to other supervised approaches, which is where we know some beginning of an answer, but we want to build up on that and let the model find more. That gets into these things like self-supervised, semi-supervised, and weakly supervised models, where you're trying to fill in gaps. And as the model learns to fill in gaps, it learns the fundamental about what's even going on in these systems. If I remove data about some of the genes, but I can fill it back in from the others, the model can begin to learn relationships for them. And so these are reasons that you have all these different levels of question with this, that it fits very nicely with what's right at the forefront of machine and deep learning methods. Because frankly, we need the methods to go after this disease, after this collection of diseases, neurodegeneration, because it hasn't boiled down to a single answer as far as we know. We found a lot of interacting answers. And so that interactingness is specifically what we want to get at in a principled way.
So I'll just kind of try to reflect that back to you and see if I got it right. It sounds, I think neurodegeneration is really interesting. Something that I find really interesting about it is that, like you're saying, it's not an acute disease state, right? People acquire this disease state over many, many years, and things in the body happen much more quickly than that on average, I would say, at a cellular level. And so you have a situation where there's many simultaneous processes and interactions in the cell that are eventually giving rise to this disease state. And because the patterns of interactions are so complicated and are happening through so many different levels of the cell's machinery, you can use AI as a way to try and find out where these interactions are happening that might be unexpected or haven't been seen before, or have been just maybe too complicated for people to hypothesize going this far. Is that right?
Yeah. I think one of the most exciting uses of AI right now is in no way to try to replace the human scientist, clinician, medical doctor, drug discoverer, or experimenter, or any of those things, but rather as a way to get a human in the loop tool that makes it possible to see things you could never have seen before. Because bluntly, with machine and deep learning methods, they're not self-guided at this time. They go after problems that we set up. And so it's on us to define what we're trying to even see. And that actually fits really nicely with the virtuous loop, where we can ask a question, get an answer, and then begin to understand how to ask better questions, and then use that to guide experiments. And then if there was something we found that we wanted to really automate, then we could set it loose on just that one automated thing. But being concrete about this, one area that I'm really interested in for machine and deep learning is the construction of landscapes. And this has always been a problem in all of the different levels of that pyramid that I was talking about. And it's a problem in science. And so in drug discovery, which is the atomistic level, when I think about small molecules, there's this problem of activity cliffs. And an activity cliff, for anyone in the space, is you have a molecule that's looking pretty promising, or maybe even have a known drug. It's in people right now. It's useful for managing one of the symptoms of Alzheimer's disease, perhaps. Maybe it interacts with dopamine receptors or muscarinic receptors. And then you add a single atom to the wrong place on that molecule, and the activity that it previously had falls right off a cliff, and it's completely ineffective and would not be a drug anymore. And yet there's this really fine boundary. You could just barely make a small change, and that can happen. And that's actually pretty common when you're exploring the chemical structures of small molecules that are known drugs or drug-like molecules. And the reason this cliff exists is that we call it a cliff and think about it as a cliff is because from the human perspective, all I did was add one atom. I added a very benign atom. I added an atom that if I were to compare the molecule before and after, it would have almost the exact same structure. And all the ways that I have similarity calculations between molecule 1 and molecule 2, it would have said this was like a 98% similar molecule. And yet it goes from exciting drug to completely ineffective molecule. What we can do with machine learning methods, one example of this is something called deep metric learning, is we can instead learn a similarity space where the machine learning approach in a landscape of, in this case, how good molecules are for that protein, can take these two structures that to the human looks very similar and more usefully move them outwards away from each other in the landscape. And conversely, other things that we would have thought are wildly different changes to it that probably I can't say anything. I have no idea if that's going to be effective as a change to my drug structure, can be brought closer together because they are effective according to the model's predictions. This landscape, this metric space, to use the jargon for it, is basically a warping, a nonlinear change to how we think about molecular structure. And so if we can construct that landscape, then as a human, I can come in and I can start exploring the landscape. I can notice that there's a cluster here and a group over here. And guess what? All of the known drugs of this category for Alzheimer's disease are over in the Southwest, or maybe that's my Southeast quadrant. And I noticed there's this one over here in a very different quadrant, but I would have otherwise thought they were all pretty similar. That one actually looks very interesting to explore. And plus there's a cluster near it that's worth looking at that I would not have thought of before. And so I begin to build up hypotheses and I can feed this in. I can go and test those. And if some of those don't work, I can tell the model, I can feed the data back in. And if they do, I feed that in as well. This is what's called an active learning loop. And it's the sort of thing that is one of the early ways that you can do AI, where you, as the scientist, as the clinician, as the pharmacist, are feeding data back in a way to update what the model knows. And over time, you can even imagine this approach moving to something called reinforcement learning, which is how self-driving cars and things like that are done. But you have to do that loop really fast for reinforcement learning. Whereas what I'm describing is, more leisurely loop where a human gets to respond, a human gets to do that. And I won't go into the details of it because I know you have a question, Brian. But you can do the same thing with images, because all the traditional ways to compare two images can really fail to tell you the images contain the same thing. We can also learn a landscape that puts all of the different types of cats, dogs, and live cell images of tau and for instance, alpha-synuclein proteins clustered in a way that comes out of the images without us as humans having to engineer the answer for that.
So to go back to this idea of cliffs that you were talking about, I mean, it almost seems like this is a situation where the human brain has limitations, right? It's a cliff because the human scientists didn't see it coming or can't understand it from what we know. But you're saying if you, one way to use these approaches for machine learning is to teach the computer to understand the landscape in a way that's more consistent with the observation that the scientist has been making. And then if the computer can help the scientist understand the landscape, then the scientist can navigate that landscape in a way that they wouldn't have been able to otherwise. They're a little bit superpowered because they have a much better map now that reflects the underlying observations that the human in the loop can make but not understand well. Is that about right?
I think so. I think the one modification I'd say is that if someone is an expert, a medicinal chemist is an absolute expert in a particular protein, then for them, that scenario I described might not have been an activity clipped in the first place. They might have expected, you know what, of course, if you add even the smallest atom there, it's going to be a disaster because that's going to poke right into the wall of the cavity, because I know that. I've studied that. I've worked with that for years. Maybe we don't even have the protein structure yet, but I've done enough discovery and I've made enough molecules and I've learned you can't add anything there. I have human intuition and expertise about that system. And so in this case, it's not that the neural network is doing something that the human couldn't have, but the neural network can do the simple version, the early version of it very fast if there's enough data for it. And that what that means is it can bring, no matter what your own background was already in the same medicinal chemistry space, it can, as you said, it can give you a superpower. It can give you an early entry, and it can give you the beginnings of intuition about the space. And now if you already have a lot, well, the type of question you can ask can be more subtle than that question I first put up there. But fundamentally, it's the sort of thing that from the programming side, we always found it really hard to do. Because even if I was talking to some medicinal chemist 20 years ago and trying to take the understandings that they had, and even if I understood them as well as I could, coming new into the system and trying to program it in, it often was going to be really mathematically complex for me to try to build that understanding into a mathematical formula and put that into a computer in a way that wasn't going to work like 3 out of 5 times, but fail in a lot of others.
There was a voice from the machine, I think.
Yeah, I think it was a little echo. Came back for a moment. We're good now. Yeah.
Okay, so I think I understand what you're saying about these methods in a sense kind of supercharging or providing more ready access to observational or kind of data processing approaches that that not everybody has access to. So maybe taking it back a couple of steps, if you've got a landscape that seems to be working well, what are the kinds of things that you do, maybe in a general description, that you do to build confidence that that landscape is correct and not trained on something that's an artifact or overtrained or something like that?
Well, fundamentally, there's 2 types of answer, and we do both. When we're ready, what we do is we take it at its word and we test it. We take some of the best predictions, we work with our colleagues, and we see if it's borne out when we try it prospectively. So we find the molecule, we buy the molecule or synthesize the molecule and test it against the protein of interest. Or we find other molecules like it and put them into the system. That's the best test because it is reality, but it is low throughput and it is very expensive. One of the things we do before that is this whole category of really, as I alluded to earlier, stress testing, or what we also call adversarial controls, where we computationally do experiments that could not be done at the actual wet lab scale. And so an example of this would be to take a model that is, as far as we can tell, succeeding and doing exactly what we asked it to do. And then we purposely set it up to fail. And we verify that it fails. Because there's a big problem, and I kind of, halfway alluded to this. There's a big problem in pattern recognition tools, which is what any of AI and machine learning currently is, which is they are rewarded, they're computationally rewarded for finding a pattern, no matter what the pattern is, no matter if the pattern is actually a biologically or chemically relevant pattern. And so there's a whole field now. So now there's a word around this. It's called shortcut learning. Did the model in your data find an artifact or part of the way the data was collected that it could exploit to get the answer that you were looking for, but that of course will not work in a real scenario? If you're doing drug discovery, did it find something out about the wells and the layout of the wells? If you're doing something in the clinical setting, is it that certain physicians are more likely to see patients who have Alzheimer's disease because they're referred to them? If you're talking, and there's classic other examples too, radiology. It works on scrambled data, that's a really bad sign because it means there was something in there that it could exploit. And there's some other flavors of that too. But I want to respect the fact that you said, overview. So we'll stick there for a minute.
All right, thanks. Okay, so maybe kind of trying to drag this into the direction of the Q&A a little bit. What's your kind of vision for the future? I mean, what's your vision for the future of your lab, the future of technology development in this space, the future for prospects for addressing Alzheimer's as a disease? Give me all your predictions.
Yeah. Well, I think Yogi Berra said it was hard to make predictions, especially about the future. But anyway, so let's see here. I hope I'm still connected as you seem to have frozen. I'm going to continue. So a prediction that we have for it, or I should say a goal that we have for the lab is with Alzheimer's disease, as I alluded to at the beginning, I don't think it's a single condition. I don't think it's a single mechanism. But what I do want to do is begin to tease that out and then find drug-like molecules or functional genomic modifications or antibodies or whatever is necessary in order for us to perturb, as in treat, that system. And so I don't expect there to be one answer, but I'd like to figure out at least what our playing field is, what specific difficult cases there are, and then move forward with each of them. And the thing that I'm really excited about is these computational methods that we're doing fit in at multiple steps in that pipeline. So one of the areas that we're going after that makes a lot of sense based on where I'm physically standing, which is the Institute for Neurodegenerative Diseases here at UCSF, led by Stanley Prusner, who discovered the prion, is we're going after some drug-like molecules that specifically interact with either the prions or the mechanisms around them, as best we can tell. And we're trying to develop molecules that will be drugs specifically for that. And so there's all kinds of hypotheses, things we want to change. We want to stop the prions from spreading from one cell to the next. And in doing this, we want to get at a core disease mechanism. Will that address all cases of Alzheimer's disease? I do not know. And it's safest to say it will not. So there's other questions that we need to figure out, which is how we even identify which are which, and how this relates to even the prions, which prion proteins are ones that we want to go after specifically. And so anyway, so boiling it down, one aspect is drug discovery. But a thing that's also an additional area that's become really important to us over time is having these tools, not just in the drug discovery pipeline, but all the way out at the other side when we think about diagnosis and also when we think about medical applications, because we have collaborations too that are outside of Alzheimer's disease only. That move into different types of cancers, which is almost like a seesaw. It seems to be maybe the mechanisms are the opposite of the mechanisms of Alzheimer's disease. And so in thinking about this, there are so many cases where we're 92% of the way there to a really powerful machine learning-based tool being used in a clinical setting. And yet the thing that stops it from being used is the fact that it hasn't really been stress tested, and it fails to an easy fix once you know to think of it in a clinical setting. Maybe it's something that looks at images of the skin for a particular type of cancer, like a melanoma, and it turns out that lighting condition matters a lot, or your skin tone, because not everyone has the same skin tone, and about fuzziness, and all the in the focal plane. These are addressable things, but models right now are overconfident, and they don't tell you when they're uncertain. And so if you are a practicing clinician, if you're in that example in dermatology, but this maps right back to neurodegeneration as well, if we're thinking about assessing someone in life for the behavioral changes around the early onset of neurodegeneration. If in these cases, the models don't tell you when they don't have a prediction, and they should be, and if they don't opt out, then if you're a clinical provider, you will learn very quickly with any model we have right now, I can't trust this, because sometimes it's wildly right, and other times it's a disaster, or I could show the same patient twice to her with a very small change, and it's a disaster. And by not having that capability to know when the model is uncertain, it completely fails in the clinical setting. So we want to be able to close that gap, that final mile problem specifically, so that in any scenario that I'm calling high stakes, be it in drug discovery, where we're going to invest time and effort and money and maybe a decade of work in trying to develop a molecule for people, or in a clinical setting, when it's someone's visit here right now and the physician can't use the tool because they can't trust it. In both those cases is what we're going after as a lab, and what we really like to change that. And so we want
it almost sounds like there's 2
tools.
problems there, right? There's figuring out how to get from what, 92, those are very precise guess, 92% of the way to 100% of the way, what's the pressure testing that's required to get there? And then also, how do you get the users of the tool to trust the tool? It seems like that's maybe kind of where the next kind of transition is going to happen in the development of these tools, right? Am I hearing you right?
Yeah, because I don't think anyone should trust a tool that they don't have reason to trust. It's trust comes from the evidence of using it and finding that it works, or at least it tells you, I don't know, I just can't address this scenario, this case. And that's a useful tool, because otherwise, if you're a physician, or if you're, again, a pharmacist, and you're looking at a regimen of medications that someone has already, and you're considering whether this additional one being added is safe, as long as the method tells you when it needs to opt out, it's useful. But otherwise, you have to fill that in yourself. You have to be the AI, or the I, on top of the AI to decide, is this a scenario that, as far as I know, it's given me good prediction so far? And that's terrible. That's just as much work sometimes, or more often. And so if someone ever, in this space especially, because there's so much interest in this space, I'll just say, if someone is ever coming to you and saying, trust it or don't, but everyone loves this, so you should trust it, that's not the right decision process. We strongly believe that all of the techniques from scientific discovery, where we test hypotheses by trying to break them, should be applied here too. And so instead of me saying, hey, this is a good model or not, or has a good score and some prediction, the real question around trust is, and where does it fail? It has to fail somewhere. Have you found where that is? In what ways have you tested for that? How have you explored that? Like what pressure points have you tried? And so that's the part that I wanted to address what you just said there, because I wouldn't want to find any better way to get people to trust it, if not for that first thing being there first. And I think trust comes from that.
Okay, and then maybe to try and wrap this up. So, I think you described, or at least your graduate work was really focused on trying to take as much data as possible and apply it to a problem, to a mode where you're now kind of borrowing techniques that really developed from another place and apply them to maybe a counterintuitively related problem space. Is the third act then, feeding back to guide machine learning method development in a way that's more appropriate for having the human as the user? Is that the third part of the play?
Yeah, it's so perhaps the part, and I'm glad you asked that, that I didn't focus on is the fact that with such wonderful tools and open source code available, what is possible for us to do is more than just use it, more than just test it. We change it to our uses, and sometimes we find that something even more parsimonious, something even simpler, even a neural network that is smaller than the giant ones that people are spending months and months at tech companies training. A much smaller one that we can train, actually is even more effective at one of the tasks we're doing. And so there's this mantra and a belief that more data is better. And that the data is fuel, it's the new gas, it's the thing that fuels AI. And of course you need sufficient data, but there's all these other questions around the diversity of the data, biases that are baked into the data, because the models, what they see is what they do. And we've seen all kinds of examples of completely unacceptable biases that come out of some public data sets. And so not only can we begin to get at those questions in a way that's focused on medicine and drug discovery, but for instance, recently we have a paper that was just accepted at Nature Machine Intelligence. And in this case, what we're able to do is build our own architecture that was actually a simplification of some public semantic segmentation models in order to do what we call trans-channel fluorescence learning, where we could take a data set from drug discovery of cellular images and use it to predict a missing biomarker, a missing channel that wasn't there years ago when that screen was done, and then use that missing information on that historical screen of hundreds of thousands of molecules and find molecules that were missed years ago in these multi-million-dollar collaborations. And yet the actual neural network was simpler than the neural network we started with. And it was changed for a different use that you wouldn't have otherwise maybe had a need to do in the literature and in the more computer science space. So because we ask slightly different questions and because we're really stuck up against those needs of if there's a bias, it's a problem. It's not good enough because everything we do is at the edge of what the model can do. That means that it's really much more obvious for us how to change the model and then feed it back. And so we're both giving the data publicly, the trained weights of the model and the code for the model out as part of that paper. And that's what we do as a lab. And so that's one of the areas that, thinking back to my computer science roots, which before I was in grad school, it's really fun to contribute back into that space where sometimes really simple or elegant solutions can really move the needle if you know the right question to ask. And we have and that gets to one of our first questions today, which was Alzheimer's disease and neurodegeneration being a space of so many really pressing questions that we're trying to figure out which techniques work, but which do we have to change to actually dig into that and fix some of the problems that are otherwise more theoretical problems in the models, but really matter to us a lot because that's the difference between finding a drug or not, or the model working for this patient here right now or not.
Well, I think I'm a believer in the open source and sharing of the information too. And who knows, maybe one day that architecture will be used to more accurately distinguish a cat from a dog.
One can always hope, Brian.
All right, let's try to do some questions with the time that's remaining. We've got one question, which is going back to the topic of overfitting when there's these very complicated mathematical relationships that the models are learning. Are there any approaches that are addressing the interpretability problem that force the system to explain itself in human terms?
Yeah. Okay. So first, the way we usually deal with overfitting formally is so you're familiar with the space, is regularization and other techniques that make it so the model can't follow one particular path through the network for most of its answers. But that's not really answering your question. I want to acknowledge that. Usually the thing that we care most about then is generalizability. And so the model might not be formally overfit to the data because it doesn't follow into that pattern where we can see that it's characteristically following this over addiction to a particular way to answer the question. But it could still completely fail to generalize into any new space beyond that. And that's the huge problem in the discovery case that we have, or the huge problem in the case where you might have a model that would give a wildly bad prediction in a clinical setting for a patient who's in some way underrepresented or historically overlooked in the data set. And these are huge, huge issues. So one way to solve this, as you're alluding to, could be to force the model to move through some layer of, or some kind of part of information that we do understand. And both, that's a good way to make sure that it's operating for our reasons, because we know from first principles that if we're going to go from a chemical structure to a phenotypic or to a whole organism outcome, probably needs to go through a protein, or at least we want to focus on those that are about proteins. And so you could build that in. So if you think of a neural network is effectively a series of layers of what mathematically are called neurons. They're not a neuron, but what they are is something that ingests information from the previous layers and then makes a decision mathematically about what it sends to the layer after it. And the most standard, what are called affine or feed-forward neural networks, the neurons are not connected to each other in the same layer. They always feed information in one way. So we know already that each layer of these neurons is an information gate. So what if we set up one of the layers of neurons such that if we're doing that drug discovery question, but we're trying to go from a chemical structure to a cellular output, that those intermediate layers of neurons, neuron 1 could be protein 1, dihydrofolate reductase. Neuron 2 could be another protein, could be whether this molecule interacts with alpha-synuclein. Through all the list of proteins that we know about so far, or at least we know what the answers would be so far for any molecule. And so what we can do then is when we train the neural network, the way that we reward the network for its training can both be whether it makes a good prediction about that phenotypic outcome that comes from the screen, but then we can also add a term or a part of its reward function to ask whether, if we did know the answer about an example training molecule, whether it also gave a high prediction to the proteins that we know that molecule interacts with. And so this is, I've heard this variously referred to as adding a rationale layer, where the proteins are the rationale that the model has to go through in order to make the prediction. And also some people have called it a concept bottleneck network, where the concept is the proteins and it's going through that bottleneck, but it has to predict the proteins okay. Some of the limitations though is it's pretty rare in a medical or in a drug discovery setting for us to have enough information to know the answers for all of those. Because there's like 2,000 or more proteins that we might want to say something about. In any given small molecule, we're lucky if we know a handful of things for a handful of protein partners, or just even a handful of assertions that it doesn't bind those proteins. Either way, for most discovery molecules. In fact, most discovery molecules, we know nothing for sure about the proteins. And so part of those answers at the concept or rationale layer are often lacking when we have the data at the final layer. So that's the big limit. It's a data problem there, absolutely. But that's how it's been done.
Cool. Okay, last question. There's a question from chat about the goal of eradicating Alzheimer's, and you've talked about how Alzheimer's is potentially a bit of a collection of diseases, and that's been recognized, I think, about cancer for a while. Do you think that Alzheimer's and other neurodegenerative diseases are at that same transition point where we're getting a sense to understand them well enough to start breaking them into different subtypes of disease?
I do think so. And it's a little more bottom up than top down. And what I mean by it is we're not yet at the stage where we can say, here's all categories of neurodegeneration. Alzheimer's disease, frontotemporal dementia, Parkinson's disease, multiple systems atrophy, and a whole range of them. And that we can then from the top down portion them all out into nice little boxes as to what the mechanisms of each one are. We're not there. And instead, it feels more of like we're getting better and better understandings of disease mechanism hypotheses. And we're coming up from that idea of the disease mechanism, of the protein in question, or of the way, a particular way that some cellular machinery fails. And then from that kind of the roots of that tree, our understanding comes up and branches into what we even sometimes thought were separate diseases. And an example of this is multiple systems atrophy, is one where you actually, which is historically very mysterious as far as this terrible neurodegenerative disease goes. And yet it seems to share a lot in common with Parkinson's disease, specifically through the mechanism of alpha-synuclein protein, and specifically there through the alpha-synuclein protein being a prion, where this protein is present in the body. It's not even mutated as far as we can tell. It's exactly the way it should be, except it adopts the wrong shape. And in adopting the wrong shape, that shape is a seed. And the seed, then when it bumps up against other perfectly otherwise normal, as far as we know, alpha-synuclein protein, it adopts that shape too, and this dysfunction spreads. And so this understanding that comes as we're getting better and better senses of different mechanisms, then can help us see how these different, and then depending on where it happens, then we can begin to see what the final disease is that comes out of it. But that mechanism is a core thing. And so maybe we can begin to eradicate that type of multiple systems atrophy. If we can figure out how to stop this misfolding from happening, or how to mitigate its effects, or how to help the rest of the cellular machinery that would usually already be mitigating it. Because statistically, this has to happen a bunch of times over your life, from when you're young to when you're old. Why is it usually handled? Why do you usually not see an effect? Why does that stop? And so that's what I mean by trying to take what we used to think of kind of monolithically, and with their own divisions, they had more to do with behavior or even location in the brain, and then come all the way down to a more atomistic and then cellular and other level to understand what could underlie and be in common. And I think there's going to be some stuff that will take longer than others in order to figure out, but at least we can begin to get more and more chunks of this and repartition it out. Because even in just molecular biology, there have been changes to our understanding. We had to reorder the numbering and count of serotonin receptors because we had it wrong the first time. And we had to change that. And so likewise here, I think we're going to get much more detailed understanding of particular major mechanisms of neurodegeneration. And then from that, see what's still left standing and go after those. But each time we get a better understanding, then we have a better way to know what we have to go after.
I appreciate you coming on and doing this conversation. I also appreciate you formulated that as a pattern recognition problem at the end there. So thanks again, Mike. It's been fun chatting.
Definitely. Thank you.
I think I must speak for everyone in the audience that not only did we obviously touch on Alzheimer's and folding proteins and all of the science, the traditional science, but I think we all got a masterclass on computational science today. So I just want to thank both of you so much for your time and energy and your impactful research, and for sharing it with us today and keeping us all curious. Dr. Mike Keiser, Dr. Brian Feng, thank you so much. And to our lovely audience, we hope that you enjoyed today's session. Everybody have a lovely afternoon. Cheers.